AP 120, Chapter 9, Topics Resting Potential, Threshold, and Action Potential. So, as I mentioned before, we have resting potential. The normal difference in electrical charge across the membrane when the neuron is at rest, when it is not carrying action potential. And that is negative 70 millivolts. It's maintained by the sodium-potassium ion pumps, constantly pumping 3 sodium out and 2 potassium in per ATP. So we want to generate a signal in our postsynaptic neuron. Well, the first thing that happens is those neurotransmitters will bind to the receptors that help form the ligand-gated sodium ion channels. When the neurotransmitters bind to the receptors, the channels open and sodium ions flood into the cell, going again from high concentration to low. Facilitated diffusion. This will disrupt the resting potential and the cell will become depolarized in that localized area, thereby making it more positive because of all the positive sodium ions coming in. So again, here is the ligand-gated ion channels. The ligand, the neurotransmitter binds. When it binds, it opens the channel, allowing sodium ions to flood in from high concentration to low. Now, you can see this depolarization on this special chart. On the left side is the charge of the neuron. On the bottom here is time, the passage of time. So we start at negative 70 millivolts, resting potential, and then say a few neurotransmitters are released and we get a slight depolarization. Now, if it's only a little bit of depolarization, then nothing will happen. In order to get a nerve impulse, an action potential produced, there must be sufficient depolarization. It must reach a threshold amount of negative 55 millivolts. So to reach threshold, the neuron must be depolarized from negative 70 millivolts to negative 55 millivolts. Now, if and only if threshold is reached, then we will generate an action potential. Action potential is the signal, the nerve impulse running down the axon. So basically, the action potential is a localized change in distribution of charge going from one part of an axon to the next part, to the next part, to the next part. And the way this happens, the way this charge moves down the axon is through the opening and closing of ion channels. So to see this on our chart, the way it would work is you have your resting potential, negative 70 millivolts. Threshold is reached from the binding of neurotransmitters, leading to massive depolarization, making the cell very positive. This then would be the action potential. And then you would have repolarization, returning the cell to its ideal resting potential. Usually a little bit of hyperpolarization where it goes to negative, but then it'll settle out and be returned to resting potential. This is an all or none response. Either threshold is reached and it happens, or nothing happens. So we reached threshold, negative 55 millivolts, and that negative 55 millivolts made it to the axon hillock. This will then cause voltage-gated sodium ion channels in the first segment of the axon to open. They will open, sodium will flood in, causing a massive depolarization. So threshold is reached, negative 55 millivolts, causing voltage-gated sodium ion channels to open and let sodium flood into the cell. So here is the beginning of the axon at rest. Here is the axon hillock. Threshold is reached because of the binding of neurotransmitters. That negative 55 millivolts then causes the first section of the axon to have its voltage-gated sodium ion channels open. Sodium ions flood into the cell, leading to that massive depolarization. Then, slightly after that, voltage-gated potassium ion channels will open, and potassium will leave the cell, going from high concentration to low. And this causes the repolarization, the potassium ions leaving that part of the axon. And often you get hyperpolarization where it becomes more negative than negative 70, but it'll readjust to resting potential. And again, this is an all or none response. Either we reached negative 55 millivolts or not. Now, again, this is a localized area. This is just the beginning part of the axon where we got depolarization. What happens is when that depolarizes, it will reach threshold and beyond and cause the next segment of the axon to have its voltage-gated sodium ion channels open. And then now you have your depolarization in the next segment of the axon. This will then trigger the segment after that to have its voltage-gated ion channels open, and that will move the action potential further down the axon, and so on and so on and so on along the full length. So again, one localized area is depolarized. It triggers the next portion of the axon, which then triggers the next portion all the way down the length of the axon. Now, the action potential cannot go up the axon. It cannot go in the opposite direction. And this is because of what's called the refractory period. So right after depolarization and repolarization, the ion distribution is messed up. There's tons of sodium inside the cell, tons of potassium outside of the cell. With the ions in the incorrect balance, this prevents another action potential from being triggered. So there's only one direction down the length of the axon that the action potential, the nerve impulse, can travel. All right, important transmembranes, proteins involved in the function of neurons. You want to know these, love these. We have the sodium-potassium ion pumps. They are important for maintaining resting potential, negative 70 millivolts. Pumping sodium out of the cell, pumping potassium into the cell. We have the ligand-gated sodium ion channels. These are only found in the postsynaptic neuron in the dendrites or cell body area. What is the ligand? Hope you knew. Neurotransmitters. So the neurotransmitters at the synapse bind to the ligand-gated sodium ion channels, letting sodium ions flood in and the region of the neuron becoming depolarized. And then we have the voltage-gated ion channels, the sodium voltage-gated sodium ion channels that cause the depolarization as sodium enters the cell, and the voltage-gated potassium ion channels needed for repolarization. This is the channels important for the action potential that moves down the length of the axon. So know these very important proteins for the function of the neuron. All right, as I mentioned before, myelination speeds up the impulse, the action potential. And the way it does that is basically the action potential jumps from node of Ranvier to node of Ranvier. It's sort of, not really, skips the myelinated area. So the only place we can have ions moving across the membranes are at the nodes. Can't do it where the myelination is. So again, the action potential is jumping from node to node, making it move down the axon even faster. All right, neurons, neurotransmitters come in different kinds, different types. Some of them are inhibitory, causing hyperpolarization, causing the postsynaptic neuron to become more negative. And others are excitatory, causing depolarization and making that part of the neuron more positive. So inhibitory signals make it more negative, make it harder to get an action potential. Excitatory signals make it more positive, make it easier to get to threshold. And of course, a good example of a neurotransmitter is acetylcholine. As we know, it is used to activate muscle tissue. Acetylcholine, that neurotransmitter, is always excitatory. Here is some different kinds of neurotransmitters as found in our textbook. You do not need to learn all of them, just the main categories. We have acetylcholine, know it, love it. We have the neurotransmitters based off of amino acids. So monoamines, parts of the amino acid, individual amino acids, and small 2 or 3 amino acids together. So those are a category of neurotransmitters. And then we also have gases. Nitric oxide actually is a neurotransmitter. That is it for this part of the lecture.